Hello and welcome to Automating the Ultrafiltration and Diafiltration Process with Inline Concentration Measurement to Reduce Cycling Time and Accelerate Process Development, presented by American Pharmaceutical Review and sponsored by Repligen. My name is Mike Auerbach. I am the Editor-in-Chief of American Pharmaceutical Review magazine, and I'll be the moderator during today's event. In this webinar, you will learn how variable path length technology placed inline enables real-time concentration measurements as a process progresses, even at high concentrations, how TFF automation enables rapid lab-scale testing with minimal manual intervention, and how the union of VPT and TFF automation enables a repeatable result insensitive to the types of variation commonly seen in mass balance systems. In addition, this event will hold a live question and answer session at the end of the presentation. To ask a question, click on the Ask a Question tab on the right side of your screen. Please take note the right side of your screen also features an overview of this webinar and more information regarding our speaker. If you have a technical question during the event, click the Test Your Connection button at the bottom of your screen. From there, you can access additional webinar support. Finally, we encourage you to use the social media widgets beneath the webinar to share with your friends and colleagues. And now, allow me to introduce today's speaker. Derek Carroll is an R&D leader in the bioprocessing industry who led the integration of lab-scale TFF automation systems with variable path length process analytics. As an R&D director at Repligen, he is responsible for the R&D teams developing automation systems and the development of novel filtration technologies. In his prior experience, Derek also worked in the medical device industry. Most recently, he held a role as a systems engineer manager at Becton Dickinson's Medication Delivery Solutions business unit, where he was responsible for R&D teams developing life-saving infusion delivery products. Derek holds more than 10 patents on products across multiple industries and has a track record of success in taking products from idea to global commercialization. He is a certified project management professional, holds a bachelor's degree in mechanical engineering from Colorado State University, and a master's degree in biomedical engineering from Cornell University. Derek, welcome to the webinar.
All right, thanks, Mike, for that introduction. So get to an agenda here. So what we'll be talking about today, some of the market needs and challenges that this product's meant to address. We'll highlight some of the system and its configuration between the two products that are coming together. We'll do a technical overview and start to get into some of the actual data. And then at the end, we'll open it up for some questions. So for the market need, we actually went through and reviewed a new product development area of interest survey. So 144 biomanufacturers and CMOs were interviewed for this, and this took place this year in 2022. And what they identified for the top 5 needs for biological manufacturing are the top ones are real-time inline product sampling and testing, automation instrumentation, continuous bioprocessing, DOE, PAT, and then analytical assays. And so when we were thinking about as an organization what kind of products we want to focus our development efforts on, we found that this really fully addresses the first two directly. So this is something that is real-time inline testing and sampling through the FlowVPX. And then it's also automation instrumentation through the pump portion, the KR2i, which automates the TFF process. And then indirectly, it kind of also addresses some of the others. We think this kind of technology will be something that can be applied to continuous bioprocessing at some point in the future. It's more focused on batch method for this initial development, but I think we could consider how this could enable continuous bioprocessing in the future. And then indirectly as well, this is process analytics, and it is an analytical instrument. So it indirectly addresses those as well. What we also looked at when we were thinking about how to develop products to address the biggest market needs is seeing the trend in the market towards high concentration MABs. So we say high concentration MAB, we're really thinking about that 100 mg per mil plus range. And we're seeing, as time has progressed, more and more of the new maps coming out onto the market are those higher concentration ones, with the real driver behind that being subcutaneous injections for at-home personal use versus needing to go in and receive some kind of infusion. So we really think that this product helps to address that manufacturing need, as it's able to measure in that range and control within that range. So in our typical TFF process that we most often see, UFDF or CDC, concentration, diafiltration, concentration, what we typically see is before the experiment is started, there's an initial protein concentration that's determined offline. That's using a traditional uv-vis type of system with a cuvette. At that point, you're able to determine an initial concentration factor that you want to reach. You set the number of dia volumes that you want to reach, and then, of course, your final concentration. Those are all indirectly measured through the use of scales doing mass balance instead of actually being able to measure inline. So there is some amount of error that is going to exist just in that process. But then once you're done based on that mass balance, you'll then take another offline sample, determine where you actually ended up. And of course, you're also introducing error when you're doing that high concentration measurement because you need to perform dilutions, and you might even need to actually rerun or perform dilution of the sample to get back to where you really want to be. What are these challenges specifically? So mass dependence, of course. So successfully scaling can be heavily dependent on accurate measurements and accurate setup. It's a fragmented process. So your data set locations will be in different places if you're looking at your TFF data versus your analytical data. It is highly variable, again, based on mass measurement variability. And then there's a big risk of error in setup, human error, volume, holdup calculations, dilution steps on the analytical side, incorrect tearing of scales. Those kinds of things can also be opportunities to introduce error that can really throw off the entire process before it gets started. So what we see as a solution to all of those challenges that we've kind of now framed are the combination of two of our existing products at Repligen. So we have the inline analytic piece, which is CTEX FlowVPX system. This is an inline uv-vis spectrometer, but it has the unique ability to vary the path length. So a product that we're already seeing a lot of use in monitoring, and then taking that and pairing it together with the Care2i system. So this is a lab scale TFF system used in downstream applications. And so now unifying them together with a common software platform, we're now able to use the FlowVPX not just as a monitoring system, but actually as a control and management system by using that data that's created and captured by the FlowVPX now with the automation system itself. So a bit more about the FlowVPX specifically. So it has very rapid data acquisition. We say it generates 12 QC grade concentration measurements in 1 minute. That takes place at 5 path lengths in 5 seconds. So it's measuring across that entire path length range and able to determine through algorithms in the software where is it in the linear range that gives you the true concentration measurement. That allows a very broad range for where concentrations can be measured. This goes all the way down to 0.1 mg per mil, all the way up to 300 mg per mil. All can be tested inline, no need for dilution. It will be able to address that through its variable path length. And then, of course, this allows for better accuracy. We don't need to introduce the potential for error with dilutions, and we're able to find that path length range for every measurement that's taken. So the science behind this is very familiar. It's still the same application of Beer-Lampert's law we would see in a traditional UV-vis spectroscopy. The difference, though, is in traditional UV-vis, your path length is locked. And so since that's a fixed value, your absorbance and your extinction coefficient are going to, or of course, also be locked. So really the only way to find yourself in the linear range of the spectrophotometer so that it's not saturated at high concentration is through dilution. And then, of course, after you perform that dilution, you find the sample with where it falls within the linear range, then you reapply that dilution factor to find your actual concentration that you're interested in. What variable path length spectroscopy allows is your concentration and really what you're actually interested in measuring is locked, and the path length becomes unlocked. So by taking advantage of the system itself and not being locked in by the geometry of a cuvette, being able to actually dynamically control that path length through use of a fibrette that is adjustable on a motor, we're now able to unlock that variable for path length. And this allows for accurate measurement and no dilution. So for every measurement that's taken, it's kind of a combination of all that data. So you take those 12 QC grade concentration measurements, you determine where it's in that linear range where it's not saturated, and then that will give you an output for a concentration that can be used to chart out and continuously collect data throughout a process. A little bit more about the KR2i. So this is our turnkey benchtop TFF system. It enables walkaway automation. There are 13 automated process control modes. Mainly that is combinations of concentration and diafiltration. There are also modes for cleaning, recovery, some other common uses of the system, flux excursions. But really, what we mainly see its use with the Flow VPX is that concentration and diafiltration mode. Robust processing, so these are highly configurable flow paths and setups for systems. So that allows processing of volumes down to 10 milliliters to as high as 10 liters. And that really helps to address the high concentration types of applications that we're looking to go after, where at the lab scale there might not be a lot of starting material and we need to have very minimal holdup volumes to reach those really high applications. We minimize risk by offering fully enclosed ProConnex custom flow paths. So we're able to work with customers to understand the specifics of their needs and develop a flow path that will fit with their use case. And then that flexibility and configuration also applies to the system itself with different process accessories, scales, sensors, whatever is needed for a particular application. And then a part of the whole Repligen solution is we also offer flat sheet cassettes and hollow fiber filters for whatever TFF need someone has. So looking at how this all comes together, so there's a picture in the top left of all the different components of the system, and then a diagram of how everything comes to be linked together. So this would be very common for any UFDF type of experiment or trial. The main difference now being that the Flow VPX is a part of this. So the Flow VPX is actually placed in line just after the feed tank. So that's the place in the circuit where it's most representative of the concentration of the full batch of material that's recirculating in the recirculation vessel. And opposed to, a common question we get, why not put it on the retentate line? That's actually going to be more concentrated material coming off of the retentate. If you think about what's happening in your TFF process, you're removing liquid from your protein through the filter. So more concentrated material is flowing back through the retentate line. So that's why we made sure to put it on the feed line where it's most representative. So how this all comes together, this is really a software project. It's all familiar hardware. It's just a matter of integrating it together. So we've developed a new software platform, which we call the CrossFlow RPM software. RPM standing for real-time process management. So it enables all of the familiar TFF automation control, but now allows that based on inline concentration. It gives real-time data every step of the filtration process, automatically generates graphs, charts, and trends all of those inputs and outputs that are crucial to understand in your process. And it's a very flexible plug-and-play technology so that you can set it up how you need it for your particular application. So now to get into what an actual run may look like. So this is a setup where we did something that's very typical UFDF run for high concentration MAB. It is a CDC. In the recipe setup, we've actually selected now the VPT device, the variable pathlength technology device. For step one, we've selected now protein concentration measured directly. In this case, we chose 38.5 mg per mil. For diafiltration mode, we're still using diavolumes in this case, so we're doing 4 diavolumes. And then the final step is our end concentration, where we really want to end up, in this case, 150 mg per mil. So again, looking now at ultrafiltration 1, so an initial C1. In this video, we start the concentration at the beginning. In this case, it's a constant feed where vacuum's drawing in additional feedstock. Our recirculation vessel is maintaining constant volume while that's being added, and concentration is going up as we start this process. We can actually see an inflection point once the feedstock bottle is drained, where concentration starts to ramp up and the bottle starts to drop all the way up until the time the concentration is actually met. Then in diafiltration mode, we're actually holding constant concentration, doing a buffer exchange. So the diafiltration pump is adding buffer to the recirculation vessel to maintain a constant level, and that concentration is maintained, kept the same throughout the run. If we were to see any kinds of anomalies where it was changing for some reason, that's something we'd be able to identify and know exactly when it happened by tracking throughout the process. Then lastly, in the final concentration, we're now concentrating up to that final target. The recirculation feed volume is getting down to very low levels. We're really taking advantage of that small holdup volume so that we can get all the way up to, in this case, 150 mg per mil. And at that point, the process will end and you are left with only your product and an accurate measurement, knowing that it's exactly where you wanted to end up. Okay, so now that we've seen how a typical run would look, wanted to show another experiment that we also did, kind of comparing the old method, mass balance, to new method, inline analytics. So these are two experiments with very similar types of endpoints and goals for what you would want to accomplish. Up top, we're using the mass balance targets. So using concentration factors representative of the values that we hope to reach. and dia volumes, diafiltration volumes, representative of the exchange of buffer that you would want to do. In the inline analytics targets, we're now taking advantage of our ability with the VPT device to set protein concentration, so we can reach exactly the set point that we want to, as we did in the last experiment. What's also being done here is for step 2 in diafiltration mode, instead of using dia volumes, we're using conductivity. So taking advantage of inline conductivity sensors that we have the ability to also use as set points. We now are fully separate from mass balance and able to control the entire process through inline analytics. So if we compare the results then between the two experiments, we see something very interesting with a lot more control. So in this graph, we have 2 axes. So we have both protein concentration and conductivity. And then in the gray, we have our results for each for mass balance. And in blue, we have our results for each for protein concentration. So what we see when we control with inline analytics for the protein concentration, we hit very close within a very small percentage value the target. So our target C1 38.5, we can see, we're in that 38.5 to, under 40 range. So, and holding that consistently through the duration of diafiltration. Then by the end, for final concentration, again, we're within just a few percentage points of the target, which is 150. That's in contrast to our mass balance, which we see over concentrating both for our initial target and also for our end target, just purely due to the fact that there is some amount of setup error, and also the fact that by the end, especially for the final concentration, when you get down to that small of a volume, your change in concentration to change in unit volume has become very amplified, where even a few milliliters can be a massive difference in the concentration values. The other interesting thing we see here related to conductivity and the diafiltration mode is we actually see that the amount of buffer exchange that we were doing, in this case 4 dia volumes, which is actually quite low for a typical process. 10 dia volumes is not uncommon. We see that a lot of that is unnecessary. So if you look at the conductivity comparing the two, you can see you had already reached the target well before diafiltration had actually ended, and all of that extra time, you've already reached your target for completing the exchange, is really unnecessary. So we see a much more accurate and also a much more efficient process through the use of these inline analytics. Then we also wanted to take a look at consistency across our different spectroscopy platforms, taking a look at what was actually measured between each of them. So we, of course, use our FlowVPX system. That's the data you just saw. We also took offline measurements with our Solo VPE system, which is an offline version of variable pathlength technology, and saw very, very good consistency between the two platforms. And then we compared that to traditional fixed path length. We also saw good consistency, but as we know, there is error in doing dilutions with that technology, but really did show good consistency across all, and that what we were measuring with the FlowVPX is accurate and usable data. All right, so that kind of concludes the data portion. So just to kind of summarize what we're offering now with this system. So it's able to strengthen process control, high quality and highly reproducible results. We're increasing process efficiency and reducing cycle time through the use of inline analytics to have the most efficient process possible. And then we're also reducing risk of something going wrong that could lead to a gross failure and end up measuring exactly what you're actually interested in getting as an output from your process. All right, thank you. Really appreciate the time, and yeah, I'd like to open it up to if there are any questions.
Thanks, Derek, for your excellent presentation. I'd like to remind everyone that you can submit any questions you have for Derek in the Q&A box on your screen. I'll get to as many as we can. There's also a resource available in the resource tab that has more information on this topic, and I encourage you to take a look at that and download that and look at that information. All right, Derek, so our first question that we have in is this technology scalable to pilot/production?
Sure. For this integration, we actually started at the lab scale. This is the first product that we're releasing for what we envision will be a full product portfolio. So at Repligen, the FlowVPX is already used for pilot and production in GMP environments. Same for our automation systems, including even the KR2i actually in some cases, but especially our larger systems are already being used at that scale. So we fully intend to continue integrating larger systems with the FlowVPX and releasing similar software in the future.
Excellent, thanks. All right, let's take a look at our next question. Can this be applied to other molecules besides monoclonal antibodies?
It can. So in this development, we were really focused on the high concentration mAb. So that's really where we focused most of our development work, did most of the testing during verification, validation. But we do see the FlowVPX used in a wide variety of biomolecules, so it can measure at different wavelengths and for different types of applications, it can definitely be used. So, and especially if you're doing similar UFDF types of processes, we expect you would have a very similar type of result as you would in the high concentration lab types of applications.
Mm-hmm. Great. Let's take a look at our next question. How can I get a demo?
Sure. So to get a demo, you can there's resources that are in this webinar. You could also reach out to your Repligen salesperson, and they could set something up for you. So basically the process is we have a number of demo systems. If there's interest and you want some help with your application, want to see if it can apply, then we'd be able to set something up and take a look if this system is right for you.
Mm-hmm. Excellent. Thanks again. Our next question is, what is the difference between this system and inline UV sensors from other vendors?
Sure. So, yeah, I mentioned in the presentation fixed path length, but maybe it was more in the context of offline measurement. There are inline UV measurement types of products that are on the market. We're pretty familiar with those, actually offer them with some of our systems. Typically, though, again, they're fixed path lengths, so can't measure up to high concentrations, don't have the variable path length ability to adjust as concentration increases. So we really see their use mainly being looking for breakthrough, that kind of thing, really detecting if a particular molecule is present or not, and not so much being able to give accurate measurements of the concentration of that molecule.
Mm-hmm. Just a reminder to everyone watching that if you have any questions for Derek, to please type them in now in the Q&A box on your screen, and we'll get to as many as we can. Having said that, our next question is, are there processes other than ultrafiltration and diafiltration for which the technology can be used?
So this system is really a TFF system, so it's more focused on that application. However, the FlowVPX is useful for measuring inline biomolecules. So there are other applications that it's used for. So in particular, we've seen it being used in inline chromatography. That's not something we offer as an integration at this point, but it is being used in that kind of application. We also see it being used in fill finish types of application, formulation application. So yeah, maybe not specific to this product, but we do see the FlowVPX, this type of technology, being used for other types of processes as well.
Okay, thanks. Moving on, our next question is, if I already have a KR2i, can it still be integrated with a FlowVPX?
Yes, it can. So yeah, we're offering that potential that actually for the kind of similar to an earlier question, for the purpose of a demo, if you already have a KR2i, we could easily just bring the FlowVPX and a tablet with the new RPM software and use your existing TFF system. Similarly, we'll offer options to, I guess, upgrade or purchase the tablet and the VPX that you are missing to enable this integration.
Mm-hmm. Thanks again for your answer. Our next question is, does this work for measuring nucleic acid concentration like A260 instead of A280?
It does, yeah. So what we do, the driver behind it, the light source, is a full spectrum uv-vis. So typically it's used at 280 for these types of protein applications, but it is full spectrum. It can be configured for 260, really anything in that entire visible light or UV spectrum. So yes, it can measure nucleic acid at 260.
Excellent. Thanks, Derek. Well, I believe those are all the questions we have today. We'd like to thank Derek Carroll for sharing his knowledge with us, and also offer a special thank you to Repligen for sponsoring today's event. Please keep a lookout for an email containing a link to view this webinar on demand and to share with your colleague. Thanks for attending the webinar today, and please enjoy the rest of your day.